We are on to chapter 16. Now, we skipped chapter 15, so we'll have to catch you up with regard to what you missed there a little bit, just so you understand where we're picking up now. But remember, chapter 14 was all about Mendel and his peas and the fact that it was finally beginning to be understood that there is something that is inherited. It is a something. They didn't know what it was, and that is expressed in traits that eventually became something called an allele, and that it is inherited as a substance that eventually started becoming called chromosomes, and that it is passed on from generation to generation and can be studied physically in how something looks, and also understood in terms of the material that gets passed on. This particular chapter is all about this DNA molecule, and first part of it is how was the DNA molecule first understood in terms of the anatomy of it. Certainly it is at this time not totally understood even yet, but that's what this particular chapter is about, is the DNA molecule itself, how it became to be understood in its anatomy, and also how does it replicate. But it's definitely, I guess, according to your book, the most celebrated molecule of our time. So that's a pretty good accolade for it, and that your information that you inherit is encoded, and it is very much of a code on your DNA that gets reproduced in body cells. And you have the famous picture of the double helix, the spiral double helix with these nitrogen bases, with a sugar phosphate backbone. So we'll learn more about not only the bases but also the sugar phosphate backbone, and how it was discovered that this is where the traits that you inherit are housed. So of course it's Watson and Crick, and if you remember, Mendel worked at around 1850 something, 1850 all the way to 1870, 80. He was working and trying to be published. Was not very well accepted in his time, and as a matter of fact, Mendel died in obscurity. It's only after his death that we have recognized him. But it's in the 50s, so about 100 years later, that Watson and Crick actually published or introduced their model of the DNA molecule. They had been working for some time, but this is when they published, and of course they won the Nobel Prize, etc. But here's a picture of Watson and Crick, and it also is an image of their of a model that they created with regard to the double helix. You can kind of see the two phosphate sugar backbone, and then you can also see the nucleotides joining together there. DNA now is generally accepted, I guess more than generally accepted, that it directs not only your biochemical life, your anatomical life, but to some extent your behavioral traits also. Now that's not as much known about DNA's effect on your behavioral traits, and certainly this is where you get into maybe the less you just get into, in some respects, the more interesting aspect of DNA, but all of that is directed by DNA. So the thing that I need to catch you up on that happened in chapter 15 that I didn't make you read, which is kind of an interesting chapter, but it's okay, I can catch you up with it. This gentleman called T.H. Morgan picked up the information that Mendel created, and he started working in about the turn of the century, about 1900s, with fruit flies. And one of the reasons why he picked fruit flies is they only have about 4 sets of chromosomes. So they have 3 autosomal and then 1 pair of sex chromosomes. So the chromosomes were pretty simple, and by that time they could actually see, they could see chromosomes on the microscope, and so they could see what they looked like. I guess you might say, when they were condensed. And what he did was he crossed, and then of course the fruit flies are also very good experimental items because they reproduce fast, they are readily available, and they can be pretty much controlled with regard to their reproduction if it's in a controlled environment. He worked in what was known as the fly room, where that was in those rooms were these fruit flies. So anyway, he bred these flies, and he did it for about 5-6 years and nothing. He was so disgusted, and then finally they had a wild fly come in, and there was one set of breeding, and so what you had was a shift in eye color, and so from the red-eyed to a wide-eyed fly. And what they found, again, because limited DNA data, is the wide-eyed were always on the males. So the wide-eyed wild fly that they brought in, or mutant fly, I guess they called it, was a female, but all of the offspring from that female that were wide-eyed were male. And what this told them was that everything was passed on, except for when the female passed on the sex chromosome. Remember, the sex chromosome for the male is an X and a Y, so the male gets the X from the female and the Y from the male, that the only thing she could pass on was a recessive gene. And so not only did they then fully understand that chromosomes are what were the inherited data, they actually understood that it was a spot on that chromosome, because the only time you had the wide-eyed males was when they were males. So that particular trait had to have been passed on in the sex chromosome that was coming from the female. And so this was kind of earth-shattering. Then what they did was they took chromosomes and looked at what is the biological composition. So what chemicals are they made up of? They're made up of DNA, deoxyribonucleic acid, and proteins that phosphate. And they had a phosphate group and then a glucose, a carbohydrate group. But what they first thought was that the protein was a much better candidate for the carrying on the inherited material, because proteins were much more complex. There were more of them. They had isolated about 20 different proteins at that time in the human, anyway, and they had only isolated about 4 nucleotide bases. So they thought, well, only 4 bases, that can't be it. So most people lean toward the protein as the area that was carrying the actual code. So the experiments that got set up with regard to further study had that in mind, that we were looking for something on the chromosomes, and that something on the chromosomes was either a nucleotide or a protein. So the next experiment had to do with a gentleman, Frederick Griffin, about I guess 10 years later. What he did was he worked with 2 strains of bacterium. One was deadly to these mice, and one was harmless. And what he did was, and so he injected the mice with each of them and found out this idea about the deadly and the harmless. But he took the deadly one and exposed it to extreme heat. Now we know that heat denatures protein. They know that. And so the heat killed the remains of the pathogenic strain. So it killed it, made it harmless. And so he took the pathogenic strain, heated it, and then injected that in the mouse, and the mouse was fine. So then he thought, oh, wait a minute, so it must be the protein. It must be the protein that's doing this. But then, for some reason, he took some of the pathogenic strain that he had that he had killed and put it and mixed it in with the living harmless strain. And he waited with that, and then he injected that into the mouse, and then suddenly the mouse died. And so what he theorized is that the killed pathogenic strain, even though it was neutralized, it actually had something in it that then could go into the living cell, henceforth the idea of a virus. They didn't know that at that time. Go into the living cell and then actually reactivate itself. These were all terms they didn't particularly know at that time, but this is what happens. Is it was actually a virus. That virus then can actually go into the living cells, take over the living cell, and then become active again in terms of pathogenic. So this made them wonder, is it the protein or is it the DNA? So then further experiments were done. These are that concept, actually, before I got ahead of myself, was considered transfiguration, because what happens is you take a virulent strain of virus, you kill it, but then you introduce it to a living cell, and then it be able to become reactivated. So this term transformation was coined in the 40s, and that led to the understanding about how viruses work, which was very, very useful. And by the way, almost all of this work is being done to try to create, at this time, to create ways to fight diseases like pneumonia, tuberculosis, diseases that were highly affecting the populations at that time. And so the next experiment worked with these phages. We've seen these phages before, and the phages, because we were still trying to figure out, is it the DNA or is it the protein? And these phages, these viruses that use bacteriophages, and you have an image here. If we go and you actually have, it's pretty amazing. You have a microscopic image of it on the left, and then on the right, with regard to this phage, this capsid that you have at the top is protein based. The DNA inside this viral genome is the DNA based, and the rest of it, the sheath and the tail fibers, are all just tools to actually get that DNA or that viral DNA into the cell. And by the way, just looking at the size here, this line you see is an E. coli cell. So this phage is incredibly small compared to the cell. You can think the cell is huge. And when we get to it, this phage, which is very small compared to a cell, is about 10 times wider than what DNA is. The actual double helix, this phage is about 10 times wider. So when we get to this double helix and we start talking about it, it was a very, very small thing, particularly when you look at how they could image it. So anyway, you have protein on the outside, you have your DNA on the inside. So you have a perfect way to study, is it the protein or is it the DNA? So what they did in 52, we've moved on to 52, Hershey and Chase, they actually performed experiments by using what's known as a T2 phage. They used radioactive protein on the capsid on the outside, and then they used a pellet, a radioactive pellet, with the DNA on the inside. Radioactive means it's going to be able to be seen. Radioactive phosphorus. So the protein is going to have sulfur, and the DNA is going to have phosphorus. So those were two chemicals they knew were different. And unfortunately, we're going to have to pick this up on the next one. They knew those two chemicals were different, and they attached the radioactive element to the phosphorus for DNA and the sulfur for protein. We'll pick this up on the next one.